Hi, my name is Jack Carter. Today I'm going to be presenting my metabolic pathways poster. As you can see up here, there is many different metabolic pathways listed. Some of them connect with each other as well. A lot of them have the same coenzymes. And up here is a little color-coded sheet to tell you which color represents which coenzyme. For today, I'm going to be starting with the glycogenesis pathway up here. As you can see, it starts with alpha-D glucose, which comes from digested glucose that enters the bloodstream and transforms into alpha-D glucose, which is the naturally abundant anomer. Alpha-D glucose then enters the cell across the plasma membrane and is phosphorylated to glucose 6-phosphate by hexokinase or glucokinase. Glucose 6-phosphate is converted into glucose 1-phosphate by phosphoglucomutase, this enzyme right here. Glucose 1-phosphate reacts with UTP to form UDP glucose, which is the activated form of glucose. From there, UDP is separated off and a single glucose unit is left here. Single glucose units are then combined to a glycogen primer. Glycogen primers are at least 4 glucose units long, and glycogenin binds those single glucose units to make the glycogen primer. Glycogen primers are then expanded into glycogen chains. For this to happen, glycogen synthase adds glucose units onto the non-reducing end of the glycogen primer or glycogen chain. From the non-reducing end, we come here into glycogenolysis. Glycogen phosphorylase cleaves off a glucose unit of the non-reducing end of glycogen by adding inorganic phosphate, producing glucose 1-phosphate right here. Glucose 1-phosphate is converted into glucose 6-phosphate by the enzyme phosphoglucomutase. Let's see. In the muscle, this glucose 6-phosphate enters glycolysis to produce energy. It could also enter glycolysis if levels are elevated. In the liver and kidney tissue, glucose 6-phosphatase removes the phosphate from glucose 6-phosphate to form free glucose right here, which can then enter the bloodstream to maintain blood glucose levels. Let's see. In here, as we enter glycolysis from glycogenolysis, phosphoglucose isomerase converts this glucose 6-phosphate into fructose 6-phosphate right here. Phosphofructokinase converts ATP to convert fructose 6-phosphate into fructose 1,6-biphosphate right there. And then we have aldolase, which splits fructose 1,6-biphosphate into 2 3-carbon molecules, or 2 3-carbon molecules, which would be glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Triphosphate isomerase converts this dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate dehydrogenase adds inorganic phosphate and forms 1,3 biphosphoglyrate while reducing NAD plus to NADH right there. Phosphoglycerate kinase transfers a phosphate from 1,3 biphosphate to ADP, producing ADP and forming 3-phosphoglycerate, which will be right here. Phosphoglycerate mutates, rearranges 3-phosphoglycerate into 2-phosphoglycerate. Enolase removes water from 2-phosphoglycerate, forming phosphophenol pyruvate, aka known as PEP. Pyruvate kinase transfers a phosphate from PEP to ADP to make ATP and produce pyruvate right there. from there, pyruvate can go into gluconeogenesis, or the TCA cycle right here. In gluconeogenesis, pyruvate, with the help of pyruvate carboxylase, transforms into oxaloacetate. Let's see, oxaloacetate uses phosphoenolpyruvate carboxykinase to transform into phosphoenolpyruvate. Let's see. And then phosphoenolpyruvate can either transform into fructose 1,6-biphosphate, or it can come back. It's a reversible reaction right here. This fructose 1,6-biphosphate, with the help of fructose 1,6-biphosphatase, reacts and goes into fructose 6-phosphate. Same as earlier, this is interchangeable. It's a two-way reaction. It can transform into glucose 6-phosphate or come back into fructose 6-phosphate. This glucose 6-phosphate, though, uses glucose 6-phosphatase and goes into glucose, which then goes into the bloodstream to maintain blood glucose levels, or it could be used for energy as well. Down here, though, for the hexose monophosphate shunt, this glucose 6-phosphate from glycolysis is used in the first step of the oxidative stage of the hexose monophosphate shunt. The glucose 6-phosphate is oxidized by glucose 6-phosphate dehydrogenase to form 6-phosphogluconolactone and produce a NADPH plus H plus. Gluconolactonase converts 6-phosphogluconolactone into 6-phosphogluconate. 6-phosphogluconate is oxidized and decarboxylated by 6-phosphogluconate dehydrogenase to form D-ribulose 5-phosphate, another NADPH plus H plus coenzyme, and a carbon dioxide molecule. Going over here, glucose 6-phosphate is converted to fructose 6-phosphate by hexose phosphate isomerase. And up here we have D-ribulose 5-phosphate from the oxidative stage being converted to D-ribose 5-phosphate for nucleotide synthesis, or deoxyribose 5-phosphate, both by phosphopentaisomerase. We have here transketolase moves a 2-carbon unit between sugars, forming glyceraldehyde 3-phosphate. Transaldolase transfers a 3-carbon unit to form fructose 6-phosphate. Transketolase again acts to produce another fructose 6-phosphate and glyceraldehyde 3-phosphate right here. Fructose 6-phosphate and glyceraldehyde 3-phosphate can re-enter the glycolysis pathway as well, be converted back to glucose 6-phosphate to go through the whole synthesis. Coming back up here to the TCA cycle, though. So, starting with pyruvate. Pyruvate is converted into acetyl-CoA for the TCA cycle by pyruvate dehydrogenase, producing NADH plus H plus coenzyme and releasing carbon dioxide as a result. So you have here oxaloacetate combines with acetyl-CoA to form citrate. Citrate is rearranged into isocitrate. Isocitrate is oxidized into alpha-ketoglutarate, releasing carbon dioxide right here and producing another NADH plus H plus coenzyme. Alpha-ketoglutarate, though, is converted to succinyl-CoA, releasing another CO2 and forming a second NADH coenzyme. Succinyl-CoA is converted to succinate, generating GTP or ATP by substrate level phosphorylation. Succinate is oxidized to fumarate, producing an FADH2. Fumarate is hydrated to malate with the addition of water molecule right there. And then malate is oxidized back into oxaloacetate, producing another NADH. All right, coming up here to fatty acid synthesis. We have here, let me see where this. Okay. All right. So, with the help of citrate lyase, ATP, CoA, and ADP plus a phosphate group coming off, we get oxaloacetate and acetyl-CoA to start out with in the fatty acid synthesis from certain intermediates from the TCA cycle, which I just listed that will come right here. These two are converted to malonyl-CoA. In that reaction, CO2 comes in. ATP energy also comes in. Acetyl-CoA carboxylase converts the ATP into ADP plus PI with the help of a biotin coenzyme as well, aka vitamin B7. Malonyl-CoA plus malic enzyme, and also an NADPH coenzyme from the hexose monophosphate shunt, then converts it into fatty acid synthase, which then can be converted into palmitate, plus any other fatty acid products, aka fatty acid synthesis. These fatty acid products are also, though, used in the beta oxidation of a saturated fatty acid, which I have up here. Starting in the first step, there's a formate. For today's whole scenario, I just drew the names of these. In my instructions, I am mentioning the structures, but also some of the information that comes along with this graphic right here. Starting with step 1, the formation of a double bond between the alpha and beta carbons. There are 4 different acetyl-CoA dehydrogenase enzymes, each specific to a range of chain lengths. The reaction also produces an FADH2, which enters the electron transport chain. Hydration reaction adds a water molecule across the double bond, forming a beta hydroxy acyl, which will be right here. The water molecule coming in. And oxidation of the beta hydroxy group to a ketone happens. The reaction also produces a NADH coenzyme, which enters the electron transport chain. Insertion of a CoA molecule and cleavage of a 2-carbon acetyl-CoA are present. Each acetyl-CoA molecule is further oxidized into the TCA cycle, which I think I have this line right here that comes into it. It comes right back to the acetyl-CoA, which I just mentioned earlier in the TCA cycle. And here we have the shortened fatty acid, or the shortened fatty acetyl-CoA is activated and then re-enters the beta oxidation pathway at the very beginning right here. Each passage through the beta oxidation pathway yields an additional FADH2, NADH plus H plus, and acetyl-CoA. Coming right down here to the urea cycle, starting with step 1, you have ammonia, NH3, combines with CO2 to form a carbamoyl phosphate, just right there, in a reaction catalyzed by mitochondrial carbamoyl phosphatase synthetase, and then N-acetyl-glutamate allosterically activates the enzyme to allow ATP binding. Carbamoyl phosphate reacts with ornithine, using ornithine transcarbamylase to form citrulline. Aspartate reacts with citrulline once it has been transported into the cytosol. The step is catalyzed by succinate synthetase. We have ATP and magnesium are required for the reaction, and arginosuccinate is formed, which is right here. Arginosuccinate is cleaved by arginosuccinase in the cytosol to form fumarate and then arginine. Urea is formed, and ornithine is reformed from the cleavage of arginine by arginase, a manganese requiring hepatic enzyme. This is where the urea is formed right here from arginase. Fumarate from the urea cycle also goes into the TCA cycle and can be used there. Let's see. Yes. All right, and here we have the fate of an amino acid carbon skeleton. Once an amine group has been removed from an amino acid, the remaining part is called the carbon skeleton, or the alpha keto acid. Certain amino acids are converted into pyruvate. Right here can be alanine, glycine, serine, cysteine, tryptophan, threonine. Other amino acids are converted into acetyl-CoA. Let me find it, which you can see right here, list of these. The acetyl-CoA can then enter the TCA cycle or be used to make ketone bodies or fatty acids. Some other amino acids are converted into acetoacetate, directly contributing to ketone body formation. Some other amino acids are converted into alpha ketoglutarate, feeding directly into the TCA cycle. Other amino acids are converted into succinyl-CoA, which continues the TCA cycle towards malate and oxaloacetate, I meant to say. Certain amino acids become fumarate, entering the cycle at the regeneration phase towards malate. And then we have aspartate and arginine being converted into oxaloacetate, which can either continue in the TCA cycle in this direction or be used for glucose for energy. Coming down here, we have the alanine glucose cycle. To start off, alanine is formed in muscle cells from transamination glutamate, which is generated from leucine transamination, and from pyruvate, which is generated from glucose oxidation via glycolysis. Alanine travels in the blood to the liver. In the liver, alanine is transaminated with alpha ketoglutarate to form pyruvate. Pyruvate can be converted back to glucose in a series of reactions. The glucose is then released from the liver into the blood for uptake by tissue such as muscle, which uses glucose for energy, as we know. The glutamate formed in the liver can be deaminated to release ammonia. This ammonia is used in the liver for urea production. Right, and that is basically the whole list of pathways I have explained for today. Here's a quick overview of my poster, real quick. Everything included. Got the answer key right here. That's all for today.